Welcome everyone to the Interagency Modeling and Analysis Group webinar series. I'm very pleased to have this webinar hosted by the Cell to Macroscale Working Group. The working group leads are Dr. Qinglong Lin and Dr. Ed Sander. And Dr. Sander will be introducing the speaker for today. So I'll give it to Ed.
Thank you, Grace. So I'd like to introduce today's webinar speaker, Jie Liang. He is the Richard and Lone Hill Professor in the Richard and Lone Hill Department of Bioengineering at the University of Illinois at Chicago. He joined UIC in 1999 as an assistant professor and was promoted to associate professor in 2003 and then to full professor in 2007. He received his BS degree in biophysics from Fudan University and his master's and PhD in biophysics from the University of Illinois at Urbana-Champaign. He has had several affiliations, including as a NSF-CISC postdoctoral research associate at the Beckman Institute and National Center for Supercomputing and its applications. He's also spent time as a visiting fellow at the NSF Institute of Mathematics and Applications at Minneapolis. He was a recipient of an NSF Career Award, and he was recently elected, well, actually not recently, but elected as a fellow of the American Institute of Medicine and Biological Engineering in 2007. Dr. Liang's research interests include systems biology, computational bioengineering, bioinformatics, and computational biophysics, especially in areas of structural bioinformatics, computational proteomics, molecular stochastic networks, and cellular pattern formation. Current projects in his lab include protein function prediction, evolution analysis, membrane protein nanodevice assembly, stochastic networks, tissue regeneration, and wound healing cell polarity and cellular pattern formation. With all that, I'd like to go ahead and welcome Dr. Jie Liang.
Thank you, Ed and Qinglong and Grace for inviting me for this opportunity to give me present our work. So I will talk about our work on multiscale modeling of spatial and temporal signaling and patterns in cell and tissues. So primarily, we are focusing on looking at a multiscale to study the general problems of how cell and tissue form patterns. We have to look at molecules and proteins inside cell in the form of various networks and how they interact with each other and give individual cells a particular behavior. So I will basically talk about two subjects that are very much related. One is some theoretical work and algorithm development in terms of studying stochastic control network of genetic circuits. How do we relate to epigenetic states and cellular fate? Then I will talk about our work on developing cell models to study cell and tissue pattern formations and the roles of chemical interactions and mechanical interactions of cell-cell and how they are controlled by intracellular networks. And I will give some examples on the growth pattern formation in epithelia, both in terms of topology and elongations, and then I will talk about our recent work on wound healing. So as we know, a lot of biological phenomena are stochastic, and the reason is because a lot of these events involve relatively small number of molecules, say micromolar or nanomolar ranges, and they are intrinsically stochastic. And then you also have these slow and fast reactions, and you have multi-stable systems. And there's also a large amount of cell-cell variations and external environment impacts. So overall, these stochastic phenomena are very important because they determine the epigenetic landscape of how different kinds of fate. For example, we can look at how virus infect host cells, and they might go through different fates, whether the virus gets cohabited with the host or there might be an outbreak. And we can also look at stem cells, where the stem cells might differentiate to different progenitor cells and differentiated cells with different probabilities. So there's a lot we can consider in terms of the stochastic roles in determining cellular functions and cellular fate. There's a very well-known foundational framework for studying these kind of stochastic problems, and that is the chemical mass equations. And I'm particularly interested in the type that's discrete. My lab has worked on these problems for quite some time, but I'd like to just spend 1 minute or two on the general issues. And it's a very simple way to look at a complicated system. So basically, in homogeneous systems, well-mixed systems, we have, say, m number of molecular species, and we have n number of chemical reactions. And then we can give the system a microstate. Basically, it's determined by the combination of copy numbers of all these different reactants and products at a particular time. And then we can further think about what is the probability of the system to be in a particular combination of copy numbers. And that is the state and the probability of the state. And we can think about the state space, which is the set of all possible combinations of copy numbers. And the probabilities that's at a particular time of all these states, and that's what we call the probabilistic landscapes. So when you have a chemical reactions, essentially it is a jump process of bringing a system to a different state by reducing the number of reactant copy numbers and increase the copy number of products. And then we have a reaction rate, which will depend on the concentration or copy numbers of the reactants. So with this, then we can write out basically what's called a discrete chemical mass equations. So essentially, we have all these different states coming to a particular state, and then the probability of flowing away to some other states, and all this can be summed up in a matrix form in a very simple formula. And this is a very powerful approach because it gives the full account of probabilities of various jumps, regardless the copy number is small or large, whether the jump is small or jump is large. And it can generate trajectories with the correct probabilities. However, it has had very little applications because the difficulties I will talk about. And then you can approximate this discrete chemical mass equation with a continuous chemical mass equations. Basically, if you approximate the state space as continuous, and you assume differences in the amount of molecules in neighboring states are very, very small, then you can write a continuous version of this. So the holy grail for doing biology is that if we can compute the time-evolving probability landscape over all possible states, then we can really do a lot of these. So a good analogy is that this is by Dan Beard. They wrote a book on these issues, and this is well known. Essentially, it's analogous to the Schrodinger equation in physics and chemistry, or the wave functions or density functions, or the partition functions in statistical mechanics. In this case, what we want to know is the landscape probability distribution and how it changes with time. Unfortunately, algorithmically, not much is known because we usually don't have good solutions beyond some simple problems. So there's a hierarchical formalisms when we can think about modeling systems biology of these networks. So at the simplest level, we can do these protein-protein interaction network. We can build graphs. And if we endow each of these proteins with a binding on and off state, then we have a Boolean equations. We can build Boolean networks. If we know a bit more, we know reaction rates, then we can build very useful ordinary differential equations that will work very well. But there are cases when the copy numbers are small, then we have to think about the stochasticities in the network. In that cases, we may have to use stochastic differential equations. But the most basic level, we may have to go to chemical mass equation. And the trade-off is that you have very high accuracy at these chemical mass equations level, but the complexity is also very high. The most widely used method for study the stochastic network is through simulation. It's a very well-known Gillespie algorithms since 1976 or 1977. And it has been widely used. But there are some issues with the simulation approach. Generally, because Gillespie algorithm is exact, it follows the probability events in the real reaction systems. Therefore, it follows the high probability event. So it will be very difficult to to study rare events that are biologically interesting. For example, cellular fates, changes and switches, and that kind of thing. It's also difficult to assess convergence. This is a general problem of Monte Carlo simulation. The recent work from Gillespie have pushed these biased Monte Carlo sampling methods that have shown great progress, and we have also done some work in developing adaptively biased sequential importance sampling methods for this. But on the other end, there are great work that has been developed in approximating Gillespie algorithms. Basic idea is that we don't update the copy numbers of reactants after a certain period, and this is the famous tau leaping algorithms. And the idea is to treat accuracy in reaction propensities for computing efficiency, so you can study large systems. So what I would like to talk about is our work on solving chemical master equation directly. And this is really without doing simulations. And unfortunately, analytical solutions is not possible generally, except for some very simple toy problems. So what you can hope for is to use direct numerical solutions. And the bottleneck problem is that you have to have a full description of the enormous discrete state space. And this is generally combinatorial, and it's not feasible beyond simple systems. So there have been great work that has been developed maybe about 10 years ago by Munsky and Kamash, and they developed these finite state projection methods. So the idea is that you truncate the full state space, and then you estimate how much error you're going to have. Unfortunately, it still have some issues. That is, it requires absorption states, so it cannot be used to study steady state probability landscape because there's leakage, it accumulates, and it's also difficult to choose specific subspace and limiting its size. So what states do you add to this truncated states is unclear. So another approach is to approximate this continuous chemical mass equations by taking the jump operators and do a Taylor expansion. This is known as Kramer-Moyal expansions, and then you drop everything beyond the second order in the Taylor expansions, and then you can do basically Fokker-Planck equations or Langevin equations. But the consequences of these approximations is largely unknown. I mean, there has been recent work. People are looking at some systems. What is the approximations? What you are sacrificing? So the general practice in studying stochastic network is first identify interesting biological problems, and then you construct a stochastic network. And then you compute solutions to the underlying chemical mass equations. You can use Gillespie algorithms, and you can use Langevin or Fokker-Planck, and you can use other assumptions like everything is Gaussian distributions. But then there's really a difficult in terms of understanding if there are two issues: is the network correct, or are we finding the correct solutions? So this is a challenging problem because to a large extent we don't know much. So we basically need a better algorithm for studying these discrete networks. For example, how do we know if we've computed the attractive basis of probabilities? Are they complete? Are their locations accurate? Are we missing some major peaks? Could be a different state of the cell that we didn't know about, maybe a different kind of disease. Or how can we quantify errors? And how can we compute especially probabilities of rare events, and those very low probability of a normal cell turning into a cancerous cell? And what are the best accuracy we can achieve given a large computer or small computer? What can you compute? So this kind of answer to this question is difficult. And you can always say experimental results provide the ultimate arbiter of all those models, but that's problematic as well because experimental results can provide answers, but they're too degenerate, meaning you cannot distinguish many different kind of possible models. And so the question is, can we do better on this? So we've worked on this for several years. This is with Dr. Youfang Zhao, who did a PhD and postdoc with me, and he's at Los Alamos National Lab, and Anna Terebus, who recently worked on this problem. So we have basically developed a finite buffer methods which gives optimal algorithm for enumeration of these microstates. Basically, the only assumption we assume is that there's a finite number of net molecules that are being synthesized, and this is based on mass conservation. So we start with the given initial conditions, and we can reach all states that reachable from an initial state will be accounted for, and we will not visit any irrelevant states, and all possible transition will be recorded, and no infeasible transition will be attempted. And this works for networks of reactions of arbitrary stoichiometries. So once the states are defined, enumerated, then we can really compute steady-state probability landscape by solving large-size linear equations. And we can also compute the time-evolving probability landscape using matrix exponentials, such as powerful methods such as the Krylov subspace method by C.G. et al. Here I want to give you one example. This is the simplest, one of the simplest systems with bistabilities, and this is the toggle switch example. You have basically two genes, A and Bs, and each of these genes make a protein product that repress each other. And this simple system actually turns out there's no general analytical solutions. And this system was first implemented in the test tube in the year of 2000 by Jim Collins. And that was the start of the synthetic biologies. And there's some very important work on how can you build approximation methods to study these toggle switches. But with our finite buffer methods, we can compute these probability landscape and how they change with time. For example, we start from a uniform distribution, so we can see how the probability landscape of the cells, of the systems at different states, and it changes with time, and eventually it reaches to a steady state. And if you can, we can start from a different initial condition. Instead of a uniform distribution, we're starting from these places where there's one big peak, one tiny peak, and we can start to see the time evolved trajectory is totally different. But because of the Markovian properties, it ended up with the same steady state distributions. So this is a very useful way we can look at not just the steady state, but the time evolving probability landscape, how the cell changes. So overall, we can see, we can model these things in a lot of details. We can see this on-off peak, off-on peak, and actually a very small on and on peak. And this is through enumeration about 1 million states for these problems. And we have extended this work, and we want to look at simple network motifs. Now with this powerful computational approaches, we can actually study the details of a lot of these stochastic networks without resorting to simulations. So for example, we've looked at a very simple feed-forward network involving just 3 nodes, and we can see the steady state probability landscape at different physiological conditions already shows multi-stabilities. And this is really interesting because it's from simple incoherent feed-forward motif. There's no feedback loops, and there's no cooperativities. And these number of peaks are controlled in different physiological conditions. So the next question is, can we go beyond simple motifs? Can we go to study larger networks? Most of the stochastic networks, you really cannot go cannot get very complicated. You maybe have 3 nodes, 4 nodes, but or you can use approximation Langevins without knowing the errors, or run Gillespies, and you can hope for the best. You really don't know whether you are getting the correct answers. So we've developed this method we called accurate chemical mass equation methods. It's expanding on the finite buffer methods. Now we use multi-finite buffers, and with optimize the state space. So the main idea is that we can decompose the reactions in a particular systems into different components, and then we use multiple buffers to equip each of these to control truncation errors. And there's a lot of theoretical work we did for analyzing the probability flux, the boundaries, and the truncation errors of this finite state space. So the idea is that we can take the chemical systems as reactions, take the reactions as nodes, and these nodes will be connected if reactions share reactants or products. Now we can construct what's called the reaction graphs. And in many cases, we can decompose these reaction graphs into independent components. And each of these components is then equipped with its own machineries for state enumeration. And this turns out can dramatically reduce the state space from n hypercube to n simplex, and this is enormous savings if your network is complex, if you involve a lot of molecular species, a lot of interactions, a lot of couplings. And even at the same maximum copy number, can make a lot of impossible work become feasible. So the basic idea is that we want to decompose this almost infinite size transition matrix in the framework of discrete chemical mass equation I talked about into these macro states based on their usage of this IBD. And then we can permute that read matrix accordingly, and then we can construct what's called a quotient matrices, replacing these sub-block matrices with just one number. And this one number can be computed exactly. And we can also compute the upper bound and the lower bound of each of these numbers. And then we can define the truncation error of the steady state probability landscape. Basically, that is, suppose you have the exact answer if you have infinite state space. If you truncate them because your computer is finite, and what is the error you're going to have? And the main results is that we have worked out the theories, and we can actually write out the upper bound of the truncation errors, and this can be computed a priori without costly trial solutions. So it's really, really useful. So we can determine, as a result, the minimum memory required for errors to be smaller than some predefined tolerance. And we can also optimize the memory allocation. So given the finite resources of, say, your clusters or your laptops, how much errors you can hope to get, and what is that minimum errors you can have. Here's an example that we did. So this is a well-known map case signaling networks. And mostly people look at a very simple version of this, but if you really want to be careful about the stochastics, you have to look at the details of these reactions. So this particular model is a 16-dimensional systems with 35 reactions, and it would not have been possible to do a full stochastic network analysis on this complicated network with, say, 60 nodes. And this is possible because using these ACME methods, we are able to reduce the state space from, say, 10 to the 16th power, which would basically be impossible, to about 6 million states, which is heavy lifting, but it's totally feasible with current computing resources. So in essence, the questions I raised earlier, how do we know if the computed attractive basins are complete, and how do we know the computed landscape is correct, and how can we quantify these errors? How can we compute the probability of rare events? All these can be answered with this ACME methods that we developed. So I want to give an example of how actually this can be used to really study at the cell levels. So this is one of the classic example starting all the way from 1970s, where people use phage lambda to study the life cycle choice problems. So basically, we have a phage, which is a virus for E. coli. When E. coli gets infected by this virus, there are two pathways. One is the lysogenic pathways. Basically, the virus gets integrated into the host genome. And the other pathways is the lytic pathways. Basically, the virus senses the environment is not good, and it started to take over the protein synthesis machineries. Basically, it synthesizes a lot of viral particles and kill the host. And this is the lytic pathways. And there is epigenetic memories passed along from offsprings, so the virus remembered what state it was in. And this was studied extensively computationally by Arkan, Ross, and McAdam in 1998. So we've built a model. Instead of using Gillespie algorithms, we want to solve underlying chemical mass equations. And we built a model, a simplified model with 13 molecular species, 51 reactions with about 7 billion microstates. And we incorporate cooperativities. And here are the basically the architecture of the network. We have 3 operator sites, each with different colors, with different binding constant to the 2 key proteins, C1 dimer and the pro-protein dimers. And they control each other's. And we can compute the probability landscape by looking at what is the effect if we have different UV dosages. This is modeled as degradation rate of C1 dimers. And we can see at certain places when the UV dosage is low, we'll have full lysogeny state. But if you increase the UV dosage, you start to see transitions from lysogeny gradually to lysis. And this can be studied in details. And we can compute the time-evolving probability landscape as well, at 1 second, 10 seconds, and so on and so forth. So in terms of this new methodologies, basically you can decompose this network into two independent burst and depth processes. And we can use all that machineries to estimate a priori what is the likely errors, and with certain computer memory sizes. And we can go all the way to like very small errors, and this can be computed exactly. So what we can learn from this is that we know the wild type of this kind of genetic circuits are built very well. You have a very deep threshold. They are very stable to UV irradiations. When the C1 degradation rate fluctuates, there's no effect on the cellular state. So it's basically a way of maintenance through these diffusible factors of epigenetic state of cells. But it's also a very efficient switch. You are entering the, if you enter the set point, then just 10% or a little bit more of changes in UV, you switch to a totally different status of the lysis state. So what's interesting is that we can start to look at the mechanism of how this thing, what is the behavior, what is the molecular determinants, which part of the switch is responsible for this. And it turns out the cooperativities between C1 dimers are one of the key factors. And we can further study what if we start to tweak the genetic circuits. For example, the wild type has these three components of different colors. If we change these different colors, and basically we're changing binding affinities of these operator sites to these two proteins, then it turns out that they will have very different behaviors. None of them will have as nice a behavior as the wild type. They are all kind of hair trigger, and all of these things are leaky. And this are exactly the same as the experimental studies by John Later, classic examples. And so there's a generally very good agreement between computational predictions and experimental results. So what I want to emphasize also is this algorithm is very important because you would not get the same result if you run Gillespie algorithm, because in many cases you get trapped in a meta states. And this is an example. We use one of the best Gillespie algorithm called the Stochastic. And it turns out if you run this for 48 hours and you still are away from the two correct solutions. And in contrast, if you use the chemical mass equation, our method, 8 hours, you will get good solutions. You get the exact solution already. And then if you use these stochastic differential equations, Langevin, in this particular case, it's really dangerous because you spend many, many days, you can never get out of the initial basins. So you're basically trapped there. So this is kind of interesting because in many cases, you just run simulations, you don't know where you are. You might be worried. You should be worrying about whether you are actually being trapped in one of the local minima. So overall, the ACME methods for accurate chemical mass equations can compute exact probability landscape for fairly large stochastic networks. And we can use that to study a lot of things, critical rare events, and we can gain mechanistic understanding. And this is the work with Dr. Youfang, who's now at Los Alamos, and Anna Terebus in the past with Zhao Lu as well. So the next topic I will talk about is now we have ways to deal with these networks. Can we use this to study cell development and tissue pattern formations? So the overall idea is similar. How can we understand the behavior of local individual cells in terms of division, orientation, cell-cell interactions, cell ECM interactions controlled by intracellular networks such as the stochastic network, and intercellular chemical and mechanical interactions. How do we see these global tissue pattern formations? So there have been many excellent existing cell models. For example, you have cellular POTS models, subcellular finite element models, center-based models, vertex models, immersed boundary frameworks, viscoelastic cell models, and so on and so forth. However, it's still challenging to simulate diverse and realistic cell shapes of different sizes, and to simulate cell growth and division, and as well as apoptosis, and especially to simulate the dynamic aspects of cell migrations to and independent of arbitrary initial conditions and boundary conditions, and also tissue fusions. All of this simulation at larger scale becomes a very challenging task. So we've been working on developing these cell models for this kind of problems. In our initial approach, we use very simple cell models. Basically, we have these cell in isolations and 2 interacting cells, as well as 3 interacting cells modeled explicitly. For those fully buried cells, they show up as these polygons. And we model simple compression forces as tensions, including cytoskeleton microfilaments, and so on and so forth. We have pressure forces, and we have cell adhesions or repulsions between cells. And this is a little bit like the soap bubble models, where you have basically different adhesions between cells with low tensions or strong tensions. And then we basically, in algorithm-wise, we specify volume changes at particular times, and then it results expansion of cell volume or shrinkage. And this is all follows Newton's laws. And we are moving these masses associated with these lattices, and we relax them until it reaches a steady state. But the key problem in terms of computing this is there are a lot of topological changes. When you have cell growths and cell may bump into each other, and they may run into 3 cells may run into each other. So you have to keep track of these topological change. And for these 2D cases, it's very well known in literature. And then, what are the possible cases? And this has to be really taken good care of. Otherwise, your simulation cannot proceed. So we've developed a method using some data structures so we can simulate this kind of a systems in very large scales. We can start from 1 single cell to 2 cells, and we can use this to study tissue development. And we can also model cell fusions, 2 cells run into each other, and also tissue fusions. And we can, in addition, model cell apoptosis. You can see this red cell on this right corner side, and in the next step, it's basically it withers and it goes away, and then we have to model the elimination of these dead cells, and this all can be computed. So one of the things it's important to distinguish is cell geometry and cell topologies. So cell geometries basically are about cell shape, area, lenses, angles, and so on and so forth. Cell topology is about connectivities. It's connectivities between different cells. And for this type of cells we're modeling in the epithelia, it's about the sidedness, the polygons, how many polygons you have. So 6-side, 5-side, and 7-side, and so on and so forth. And this is work by Hamad Navid and Inzi Li. Hamad Navid is now at the Toyota Institute, is an independent PI, and Inzi just obtained her PhD degrees. So one of the very interesting things you look at this is that there's a characteristic topological distributions of epithelia cell across different species. For example, you have almost identical polygon distributions between cucumber epidermis versus Drosophila larval wind disk epitheliums, and they have the same hexagon dominating distributions. So there's a lot of interesting questions about this. What is the origin of this kind of cell topology distributions? There has been theoretical works based on Markov models. Unfortunately, it doesn't provide a mechanistical consideration of biological or physical properties. And there's no quantitative analysis of this differential proliferations. And there's also very nice work by the Jieling group in Max Planck, where the Fahadifa's very influential papers where they studied the formation. So the results are quite close. The experimental results are hexagon dominating, and the simulation showed was the pentagon dominating. The pattern, overall pattern, is similar, but it's still not quite the same as experimental data. And there are topological models people carried out for studying this problem as well. So what we decided to do is that we want to model these mechanical forces, and that we want to simulate the process of cell growth, cell division, and cell rearrangement. So we particularly look at the effect of different kinds of division plane, whether it's random or orthogonal to the previous generations, or we divide the cells along the largest side by reduce the tension. So with this, we can look at the effects. So if we color each of these cells by their n-sidedness, then we can count at the end of simulations under different kind of division plane schemes. Are we seeing different distributions in terms of this different colored epithelia cells? So you can see we start from 2 cells, and then we generate a population of cells under the scheme of orthogonal division planes. And each of these different colored cell means a different end side. And in the end, we can collect thousands of cells and count how many of cell types we have for each colors. And we can also do this using a different scheme that is dividing cells along the largest side. So we can collect the different colored cells at the end of simulations, and it turns out we are able to actually simulate the exactly experimental observed distribution. It's indeed now hexagon dominated with pentagons, and there are some differences depending on particular different schemes. So this overall is very encouraging. That means we are getting the key fundamental aspects of these epithelia correct. So the next problem we looked at was a very interesting experimental observation, again by Gibson and colleagues, and that is if you look at clones of these proliferating cells that are implanted onto the bed of quiescent cells, those at the boundaries tend to have a very small number of sidedness. So the question is, does the reduction in the number of sides result from differential proliferation? So we again use this model to start from 2 cells after 12 generations to reach to a population about 4,000 cells. And then we can mutate the inner part of the cells so they are not proliferating. Now we can see what are the topological properties. And it turns out that we are able to see this reduced sidedness, but it turns out the local differential proliferation alone is insufficient. You have to have also increased the surface tension on the boundaries along with the differential proliferation to reproduce this reduced sidedness. So overall, this means the mechanical forces are very important for regulating these cell populations. So another study we did was to look at the tissue elongation of Drosophila wings. So this is a very simple epithelia. It's a classical development model. It grows from 30 cells to 50,000 cells. So tissue elongation along the PD axis. And there's a very interesting observation. There's 1 to 2 generations of cell proliferation without cell growth. Basically, you have to maintain the same total cell area, but divide cells without any growth of individual cells. So we use the same methodologies. We're able to simulate the elongation of these epithelia and with a very similar elongation factors. And the results, it turns out, the oriented cell division, that directional Q can lead to elongations, but not efficiently enough. Oriented mechanical forces can also induce these elongations. But what our novel finding, we're not aware of any experimental results or computational results, but this will be our prediction. That is, this 1 to 2 generation of non-growth proliferation, meaning cell division with reduced size, actually are quite important for this very efficient elongations. So this is a prediction that will be interesting if experiments can be done to verify it or refute this prediction. So we've now improved our cell models, and we've now developed what's called the dynamic finite element models of the cell. So essentially, we can divide the cell into 20, 30, or maybe even more of these nodes, and then we use partition of these cells into these triangular meshes, and they have internal forces and dynamics. And we model stiffness and elasticities of these cells, and we incorporate tension force, pressures, contractile forces, and cell-cell adhesions, and cell ACMs, and that's still in progress. And we can model growth and divisions, and migrations, and apoptosis, and shrinkage of the cell. And this is a student in my lab, Jieling, Zhao, and working with Youfang, Zhao, and Hamad Navid on this result. So with that new tools, now we can start to study a more complicated process of wound healing. So we're interested to look at the human skin wound healing. It's a very rapid process of restoring skin structures, and this is very critical for healing the wound bed. It's a complex process involving a lot of cell types and a lot of paracrines and autocrine signalings. And it's interesting also for cancer biology because there's a connection between chronic wound healing and cancer development. However, it's not clear how these wound healing are coordinated, regulated through these signaling inside cells or cell-cell communications, mechanical forces, chemical gradients, and we believe these multi-scale computational models can be quite useful for this model. So essentially, wound healing process is a complicated process, and this is a collaboration with Professor Luisa DiPietro, who's the head of the Wound Healing Center at UIC in the College of Dentistry. So we are looking at this multiple phases of wound healing. There's a blood clot formation, which is a recovery from hemostasis. It's a very fast process, and there's inflammation for cleansing of bacteria and foreign particles. But we're mainly focused on re-epithelializations and granulation tissue formations, and we will not address the issue of neovascularization and ECM remodeling. Those are much longer terms. So we focus on these two processes, and with a very simple minimalistic models, we incorporate primarily the fibroblasts and keratinocytes, and we are experimenting with the models of macrophages, and we have also fibrin clots and extracellular ECMs all discretized into our simulation schemes, and we have simple models of adhesions between these different cell types. So we have put these networks of protein-protein interactions into cells. Basically, we have synthesis, degradations, activate proliferations and migrations, and so on and so forth. Essentially, the key players are PTGF. It's formed from degranulation of platelets reserved in clot, and from synthesis of macrophages infiltrated into clot at early stages. And once you have PTGF, it activates fibroblast and initiates proliferations and fibroblast migrations. And it also activates TGF beta-1 productions from fibroblast. And TGF beta-1 in turns promotes collagen productions in the fibroblast. And fibroblast itself produces KGF to activate the keratinocytes, which then migrate over the granulation tissues, and they produce interleukin-1 to downregulate the fibroblast collagen production activity. So it's quite complicated networks of these interactions. So our idea is that with our finite element models of individual cells, we want to embed each cells with these networks inside them. And then so each cell's behavior is controlled by intercellular networks as well as the chemical gradients of all the cytokines secreted by the neighbors and the diffusions, and as well as the mechanical forces interactions between cells. And we are also modeling cells and ECM as well. So with this, then we are able to really simulate explicitly these processes of wound healing. So one of the key things we are able to do is to model the dynamics process of the cell migration, in this case fibroblast migrations. So basically, it's we can't just move these cells around. We have to model the process of cleaving and degrading collagens and the clot elements. And ahead of them, you can see these voids in white colors. And then keratinocytes are in blue colors. And this green colors are the clots. They will migrate by cleaving and degrading these fibrin clots and gradually move into the wound bed to start the healing process. So here we have to model the fibroblast that synthesizes new collagens, which are dropped behind the migrating collagens at the rear end. So this is all modeled explicitly. And individual cell migrations, we have to model the process of cell protrusions and the breakage of adhesions, and all this is modeled through these dynamic finite element methods. So the particular system we're studying is quite challenging. So basically, we have a dermis of 1.2 millimeters and with a length of 2.5 millimeters, and this is almost realistic. And so in this particular simulation, I'm showing we have keratinocytes, fibroblasts, and ECM elements in gray color, and as well as the fibrin clots elements. So we can see with all those initial conditions, we can see under the wound initiation control of PDGF, we see these fibroblasts starting to migrate to the wound, and it generates a lot of these cytokines, and the keratinocytes start to migrate into the wound and start to heal the covered wound bed. So this is each of these cells are modeled with embedded networks, and we can start to look at also the secreted cytokines, KGF, its concentration, how it changes with time, as well as the TGF-beta, how it changes with time. So basically, we can look at all these different molecular chemicals, their gradients, and how it influences the cell migrations of fibroblasts, how they rush to the wound side, and how the keratinocytes migrate and clean and heal the wound. So we can then look at how this closure of wound occurs under various conditions. Does it take 1 day, take 2 days, 3 days, and 4 days? And you can see roughly it's about 96 hours, 100 hours, 110 hours before the wound is completely closed. And we can also study the effects of different chemokines, PDGF synthesis rate. So if we have reduced the PDGF synthesis rate, then we can see healing still occurs, but it's mostly through re-epithelializations. There's very thin epidermises. But normal healings, you have both granulations and re-epithelializations. But if you have overactivated the synthesis rate of PDGFs, then you have too much collagen synthesized, and then you start to get very thick epidermises. You basically have what's called the hypertrophic scars in the tissue. So overall, this is a very interesting simulations that we are able to carry out. So the idea is that we want to build these multi-scale computational models of cell and tissue, and with cells controlled by intracellular networks, for example, in stochastic network or ODE networks, and how they control the diffusion of various cytokines and in interaction with the ECM with this mechanical forces modeled. So overall, we believe this approach of looking at multiple levels, looking at proteins and their interactions in the genetic circuits, as well as the phenotypes of individual cell in terms of the probability landscapes and how they affect the cell and tissue is a very useful approach. And we've looked at the particular examples of a few number of epithelia pattern formation as well as skin wound healings. And this is a joint work with Dr. Youfang Zhao, now at Los Alamos, and Yingzi Li, who's an exchange student who worked in my lab for 2 years, and Hamad Navid, who is now assistant professor at the Toyota Institute, and as well as Anna Terebus. She looked at the motif problems as well as we worked on the multi-buffer algorithms. And Jieling was the student who developed the finite-element dynamic cell models. And this is a close collaboration with Luisa DiPietro at UIC for wound healing, as well as Lisa Xu at Shanghai Jiao Tong Universities on these epithelia pattern formations. And I would like to thank funding agencies and thank you for your patience.
Thank you, Dr. Lian. If everyone can unmute their mics and their video cameras, we can start the discussion.
Hi, Ed. I see half of you.
Oh, yeah. We made it. Sorry about the confusion.
No problem.
So that was really, I was very impressed with that. I thought that was great.
Well, thank you.
I had a very detailed kind of question. If you could maybe elaborate a little more on how you were coupling the cell-cell interactions with the cell matrix interactions in the wound healing model.
Yeah, so basically, it's this work. It's through different the adhesion for adhesion coefficients. These two different types of cell and ECM, and we can model different stiffness of ECM, and we're basically incorporate that into the simulations. So there's a lot of going on in ECM, but right now we're just modeling the past materials, but it can be additional complexity can be introduced, but we don't know where to start yet.
So do you create new nodes, for example, on your cells when they contact another cell to form a connection, or do they sort of like have them get close and kind of jump to those positions? How does, I mean, I know it's a very specific detail, but I'm curious how you...
So basically we have cells, basically, before they were touching one another, they grow, and there's a topological instant that they start to be connected. And then they have these interactions with them. And then after that, then it's the mechanical forces of multiple cells interacting with that.
Okay, thank you.
Dr. Leon, you have to unmute your video camera to show yourself. Anthony had muted everyone during your talk.
Okay, yeah, okay. All right.
And the same goes for everyone else on the line. Your microphones may be muted, so you have to unmute yourself as well as your cameras.
So this is a pretty, still, we talked to our experimental colleagues. They all say it's a great model, but it's simplistic. So I think there's two ways of doing this. One is to model every possible things we can all in the model, cell model, and try to see what comes. Another model is really think carefully to selectively model just a subset of things that are the major player for particular problems. So I think each approach has a different merit. We are experimenting probably with the simple embedded simple network at this stage.
So what are your next steps for your model, Dr. Lian?
So we want to basically start to look at more different kind of problems, and one of the things is we are working with colleagues to start to model detail the physical forces. We want to get the physical forces every step. So calibrating our models with the one cell, two cells interactions, and this ongoing. And then with that, go ahead.
I was going to say, do you envision starting to actually explicitly model the cytoskeleton in addition to the chemical networks?
So we experimented with that a little bit. So in the past, we toyed with the issue of actually put some heterogeneity in these little elements, finite elements in our models at that level, maybe, but not the explicit process of it. But for ECM, it's multiple fibers to apply that in the past.
So I had another question, actually. I mean, I have tons of questions that I'll maybe talk to you separately at a later point to get some of the details. But one of the other questions I had was about some of the results you had with the sidedness of the epithelial layers. And I was curious. It looked like you were looking at the relationship between sidedness and growing, developing epithelia. I was wondering if some of that ties into some questions that exist on how cells regulate size that are related to, say, ideas about osmotic pressure and things like that. Is any of that work kind of tie into that?
Yeah, it's a great question. We toyed with the problems. This is published in our 2015+1 paper. We look at an extremely simple model of size control. So basically, it's contact inhibition, that kind of thing. And you are able to see some size control issues, but I think it really needs to go into a physical details of whether it's osmotic pressures or some other factors. But a simple inhibition of cell growth is what's needed, but the details really needs to be worked out. So it's a great question, yeah.
Jacob, did you want to voice your question?
Yeah, I was looking at the simulations, and there are some elements like finite elements that are more deterministic, but many of them are stochastic. I'm trying to figure out how much is there determinism and how much stochasticity. For example, is there any variation in the last few videos that you showed? If you repeat the same simulation several times, do you get some variation, and how much, if at all?
Your voice was breaking up a little bit, so I couldn't completely hear. Can you repeat? Maybe I'll try this again.
I'll try it again, although the question is in the chat. How much variation is there between simulations? Are they all stochastic, or are they somewhat deterministic with very little variation, if at all?
It's stochastic because of the cellular fate, like whether it's growth or whether it's moving, how it behaves, that's controlled by a stochastic model. So it's not as deterministic. You won't get the same results every time. Every simulation gets slightly different results, but it's built into the stochastic model.
But you do have deterministic elements. You noticed you mentioned finite elements somewhere there.
Yeah, so the network right now embedded in each cell, it's this deterministic model. It's ODE model. We haven't added any noise to it yet. No. But it's quite an expensive computation that I envision it will be possible down the road, with maybe just the selected elements of it.
And how much variation do you see eventually? Like the movies you showed at the end, if you repeat them many times, is there a lot of variation, or is behavior mostly the same?
Yeah, there is quite a bit of variations. Actually, we are able to run like 10, 20 simulations, and then there's a distributions, but it's well behaved. It's not like going wild. So you have an average, you have a standard deviation. It's reasonable. Yeah.
George, do you have a question? I think you're muted. You have to unmute your mic, George.
I have a question.
Hey, George.
So I work on PD, so I'm really impressed with the results that you showed. But you're not really solving any stochastic PDs. You're solving stochastic ODEs and whenever a stochastic simulation, and you're putting white noise, right? So you treat everything as independent processes, which is not right. When you in any of the results that you show, you do have these dependencies, partially correlated stochastic processes.
Yeah,
so
absolutely. Yeah.
The secret, maybe you can reveal here. How do you do that?
Okay, yeah, great question. So actually, the interesting thing is the stochastic networks, they're all coupled. There's a lot of interactions, but what's independent of what you can do is to look at the birth and death molecules. So the synthesis of certain type of molecules. So it's like a currency you introduce into the systems. This currency gets different forms, synthesized into different forms, going to different places. But it turns out, if you constructed the graph, not the normal graph of reactions, but take a reaction as a node and take these currencies as an edge and connecting different reactions, actually you find that there are independent components. So there's only some molecules can be converted to a bunch of other molecules, but they don't talk to each other. There's an aspect. It turns out.
But on that too, I actually have I need to study your paper on the master equation. But every time you have a dense system, every time you condense degrees out of that system,
yeah, they
leave an imprint. That's called memory. So the equation itself, the chemical master equation, you have memory terms there to account for the missing degrees of freedom. Yeah. Because at one point you said I have error bounds and so on, but how do you take into account that?
Great question. So the bounds are for the steady state probability. So the time evolving probably it's too hard. It's impossible. But for the steady state, we can do it. But the idea is that it's not just that error, but also how you enumerate the states. So with that independent the burst depth components, we basically proved you can replace n hypercube state space into n simplex. It's huge dramatic reductions in that. This is all provable.
Yeah, thank you.
Yeah. So I talked about two issues. One is this network, and one is this growth cell pattern. I think there's a lot of work to do to bridge these two. One interesting question, for example, is when to use this chemical master equations, when to use ODE models, when to use Langevin models. I mean, it's like you have to stitch different kind of things together and how to do it consistently. And what is appropriate to use master equation, when it's appropriate to use ODE. Lots of great questions, and I think it will be this kind of hybrid models and the integrated model. It has a great future. I think we don't know how to do this yet, but we're working on this situation.
Seems like there are no more questions from the audience.
We give a virtual hand to the speaker.
Very nice talk.
Jim Luca posted a question. Jim, do you want to voice your question? You have to unmute your mic. His question is, any thoughts on how to describe these multi-cell models in a systematic and shareable way?
Say it again.
Any thoughts on how to describe these multi-cell models in a systematic and shareable way?
How to apply multi-scale methods in a systematic way and
how to describe these multi-cell models in a systematic and shareable way?
Okay, great question. So this is really, I don't know any shortcuts. We have been talking to our experimental collaborators very frequently to have ideas how to model set parameters. What's the right adhesion coefficient? What's the right constant? So in essence, I think it's in a lot of it is known, but a lot of it is unknown. And one of the problem is if you want to set up this kind of multi-scale cell models, there are so many parameters which may never be known because you can do some in vitro measurements, and then there may be arguments to say in vivo values may be totally different, so we really don't know. But I think there's maybe good news in this, in the sense that all these biological systems are quite robust. I think if we have good guesstimate of what these parameters are, we probably will be okay. In essence, I think we will just go ahead and what's reasonable. In terms of sharing, I think it's really can benefit from the community, the collective thoughts of what will be the right behavior to model. So what is the interesting thing about epithelia? What should we model? I mean, this is really a good question, and I think some collaborations, maybe with the help of NIH, could be very useful.
Okay, I think that's all the questions.
I'll throw one more as I'm interested in the work that you do is eventually going to get from in silico to the bedside. Okay, what are some of the implications or things that could be solved by understanding what you're able to discover with your model?
So yeah, that's a great question. So for example, I think there's a great hope that we can do something useful bedside as well. For example, the the wound healing part. I mean, how do you control the concentration of these cytokines, and how do you administer different regime, and how do you do this? I mean, we can predict the different effects. I mean, the scar formation was already useful to our colleagues, and they encourage us to explore more. So I'm hopeful we're not there yet to build some model that can predict if the problem is well defined. We can actually help the clinicians or experimental biologists to search the right players and look for the right range of interactions and strengths of inhibitions and so on and so forth. So I'm optimistic.
So is there potential, my daughter does neonatal congenital heart surgery. And on the second surgery, hours are spent disconnecting scar tissue from the first surgery. Are there implications in what the model reveals for controlling the and reducing that kind of undesirable scarring?
I think those are great questions. I mean, like we certainly can model this, but probably we'll need folks like Ed to really help to really see what's really experimentally going on. We can put some model parameters, but we may not be correct, and we may not be missing some key elements. I think if Ed gets interested in those kind of problems, and maybe he can measure some key changes in the processes, and then we can put that into our model. Right now, we have limited knowledge about what's going on, but we can make some predictions based on that. But I think it's a combination of both experiments and simulation that fits each other. I think there's good hope.
Thank you.
Thank you.
I think in the UO1 investigators, there's a little group now of wound healing investigators. You may want to get together and talk.
Okay. Yeah. So I looked back at the webinar for the call. I will study that, and I think it's a good opportunity for us.
Ed, Qinglong, any final thoughts, questions, comments? I'm sorry. I think I muted you. You had some noise on the line. Go ahead.
Yeah, I don't have anything else to say. I thought it was great.
Yeah, very nice talk.
Thank you
very much. Yeah.
Thank you.
Okay, well, thank you, everyone, for joining, and I will join the call. Thank you, Dr. Leon.
Thank you very much. Take care.
Bye.